Hi, my name is and I'm researching in the Environmental Engineering Lab with Dr. Adam Smith and mentors Philip Wang and Bianca Costa, alongside my lab partner Joseph Chen. Antibiotic resistance is a threat to public health. Wastewater treatment plants receive waste with a considerable amount of antibiotics, which could contribute to the selection and proliferation of antibiotic-resistant bacteria in the environment. Throughout the treatment process, bacteria present in these technologies are exposed to antibiotic loading, which could lead to the development of antibiotic-resistant bacteria and the proliferation of antibiotic-resistant genes. While antibiotic loading in wastewater treatment systems has been studied, little is known about the bacterial communities upon antibiotic loading, specifically in emerging technologies such as anaerobic membrane bioreactors, or ANMBRs for short. Understanding the makeup of wastewater microbial communities could help assess the risks of ANMBRs as a potential biotechnology for mitigating antibiotic resistance propagation. Furthermore, it can inform which bacterial communities thrive under antibiotic loading and could help us understand the implications of its proliferation in the environment. To understand these implications, an ANMBR was operated under two conditions, one with only synthetic wastewater and another with synthetic wastewater with antibiotics. DNA extracts were taken under both conditions from the biosolid and effluent. The V4 region of the 16S rRNA gene was amplified using polymerase chain reaction, or PCR. Next, Illumina MiSeq was used to sequence the amplicons. Then, a bioinformatics tool called Mother was used to process the sequencing data. The Mother processing included aligning the sequences, cleaning the data of errors due to sequencing and alignment, clustering the sequences on an OTU taxonomy level, classifying the OTUs referencing the SILVA database, and analyzing the community structure. To visualize the sequencing depth of the sequencer used, a rarefaction curve was plotted. From the rarefaction curve, the effluent samples were better represented and had less OTUs than the biosolid samples. To visualize the beta diversity of the samples, a principal coordinate analysis, or PCOA, was performed and plotted. From the PCOA plot, distinct microbial communities were visible between the biosolid and effluent. Upon antibiotic loading, the biosolid samples remained similar to each other, whereas the effluent samples showed significant divergence. To visualize the community structure by phyla and genera, relative abundance graphs were plotted. The relative abundance graph by phyla showed that the biosolid had more unclassified bacteria and a greater abundance of minor phyla than the effluent. Upon antibiotic loading, an increase in lower abundant phyla was observed in both the biosolid and effluent communities, as well as a decrease in proteobacteria, specifically in the effluent sample. In the relative abundance graph by genera, biosolids showed a greater diversity and presence of less abundant genera than the effluent sample. Upon antibiotic loading, community structure remained unchanged for the biosolid sample. However, for the effluent sample, the community saw an increase in unclassified bacteria and less abundant genera. Going into the SHINE program, I already knew I wanted to go into a field related to biology, as it was my favorite subject and something that really interested me. I hadn't really thought much about the environmental aspect of engineering, but after learning more in depth about some of the problems that we have in society pertaining to water, I grew fascinated with it. Not only was it something that appealed to me, but I realized how closely it aligned with my interest in biology. I added AP Environmental Science and AP Chemistry to my schedule since this SHINE experience made me curious about the other aspects of our environment. Now, when I think about what I want to do in the future, environmental engineering is something I consider. I would like to give thanks to everyone who guided me along this SHINE experience. First of all, I would like to thank Bianca Costa and Philip Wang for being awesome mentors in leading me through the processes and methods of analyzing data. Next, I want to thank Dr. Smith, Dr. Mills, and the rest of the SHINE staff for providing me with this amazing opportunity at SHINE. Lastly, I would like to thank my lab partner, Joseph Chen, as well as the rest of the SHINE cohort for making these past 7 weeks so memorable. Thank you.